Okay friends, in previous lecture we are discuss about the entire informations about the most important cell organelle that is called as the nucleus. Actual nucleus ज्या ठिकाणी एंड झाला त्या ठिकाणी हा टॉपिक आपला कंप्लीट झालेला आहे. एक स्मॉल पार्ट्स त्या ठिकाणी दिलेला आहे पहा, that is known as the microbodies. So what is a microbodies? Microbodies is also a small membrane bounded minute vesicles present inside the cytoplasm of eukaryotic cell, that is known as the microbodies. पहा शब्दाचंच काय नाव दिलेलं आहे, microbodies. bodies means इथं शब्द लक्षात ठेवायचा आहे, the minute small vesicle like structure and that vesicles which enclose the different types of enzymes or different types of chemical and they are scatteredly present inside the cytoplasm. Now that's why the name is known as the microbodies. पहा अशा काही स्मॉल सेल ऑर्गॅनिल्स आहेत ज्यांना मेम्ब्रेन आहे आणि मेम्ब्रेन बाउंडेड ऑर्गॅनिल्स मधील त्यांची साईज मायन्यूट असल्यामुळं किंवा स्मॉल असल्यामुळं टर्म काय म्हणतो आपण, मायक्रोबॉडीज. अँड दीज आर स्कॅटर्डली प्रेझेंट इनसाइड द सायटोप्लाझम ऑफ बोथ प्लांट अँड animal cell. Such type of cell organelle that is called like the microbodies. As compared to eukaryotic cell मध्ये पाहिलं आपण लार्जेस्ट सेल ऑर्गेनेल किंवा लार्जेस्ट साइज्ड न्यूक्लियर सेल ऑर्गेनेल म्हटलं, न्यूक्लियस आहे, माइटोकॉन्ड्रिया आहे, क्लोरोप्लास्ट आहे, व्हॅक्युल्स आहेत, एंडोप्लाझ्मिक रेटिक्युल्स आहेत. these are the large cell organelle. Similar like that the some minute vesicles which include the different types of enzymes required for the various metabolic activities. Such minute vesicle scatteredly present inside the cytoplasm, that is known as the microbodies. So these are present in both plant and animal cell. Now these microbodies which include the main three types. यांच्यामध्ये मायक्रोबॉडीज मधील पुढे आपल्याला रेफरन्सेस येणार आहेत, पण तीन टाइप्स लक्षात ठेवा. एकाला म्हणतो आम्ही that is called like the peroxisomes. So second one is also known as the glyoxysomes and third one is known as the spherosomes. So all these are three collectively known as the microbodies. So how many types of microbodies? Main three types of microbodies are present inside the cytoplasm. First one is the peroxisome. So peroxisome is the microbodies, glyoxysome is the type of microbodies, spherosomes these are the types of microbodies. Out of these the peroxisomes which is present in both plants and animals. प्लांट्स अँड ॲनिमल युकॅरियोटिक सेलमध्ये पाहिलं तर कोणता या ठिकाणी मायक्रोबॉडीज प्रेझेंट असते, दॅट इज कॉल्ड लाईक द peroxisomes. And remaining two, these are present inside the only plant cell. ओनली कुणामध्ये प्रेझेंट असते, प्लांट सेल. सो जिथं जिथं एक्झाम्पल येईल तर त्या ठिकाणी शब्द लक्षात ठेवायचा आहे. आउट ऑफ दिस फर्स्ट वन इज द peroxisome. दोन कॅरेक्टर लक्षात ठेवा. द peroxisome प्रेझेंट इनसाइड द मेसोफिल सेल ऑफ लीफ along with the chloroplast and mitochondria which perform one of the process that is known as the photorespiration. ॲक्च्युअली फोटो रेस्पायरेशन प्रोसेस आपण डिटेलमध्ये पाहणार आहेत फोटोसिंथेसिसमध्ये, पण ॲक्च्युअल आपल्याला माहिती आहे की फोटोसिंथेसिसचं स्ट्रक्चरल अँड फंक्शनल युनिट कोण आहे, क्लोरोप्लास्ट. करेक्ट. फर्स्ट सेल ऑर्गेनेल पाहिलं आपण, काय म्हटलं आपण, क्लोरोप्लास्ट. सो दिस क्लोरोप्लास्ट विच इज प्रेझेंट इन कोणत्या सेलमध्ये असतो, मेसोफिल सेल. कोणत्या सेलमध्ये आहे, मेसोफिल सेल. ॲक्च्युअली मेसोफिल सेल्स कुठं असतात, कोणत्याही लीफचा जर आपण सेक्शन्स पाहिला तर या लीफला आउटरमोस्ट जो लेयर असतो त्याला आपण काय म्हणतो, एपिडर्मिस. काय म्हणायचं त्याला, एपिडर्मिस. सो दॅट इज कॉल्ड लाईक द एपिडर्मिस. And another one is the lowermost parts, so that is called like again epidermis. यालाही काय म्हणतो आपण, एपिडर्मिस. तर इथं शब्दाचा अर्थ लक्षात ठेवा, दोज लेयर विच इज प्रेझेंट टुवर्ड द आउटर साइड द नेम इज नोन एज द एपिडर्मिस लेयर, बट द प्रेझेंट टुवर्ड द अप्पर साइड म्हणून याला म्हणतो आम्ही अप्पर एपिडर्मिस. आणि जो लोअर साइडला आहे यांना म्हणायचं लोअर एपिडर्मिस. लीफचा सेक्शन्स घेतल्यानंतर लीफमध्ये जर आपण पाहिलं अप्पर अँड लोअर एपिडर्मिसच्या मध्ये या ज्या सेल्स असतात या सेल्सला एक नाव दिलं जातं, दॅट सेल्स इज नोन ॲज द, कोणत्या सेल म्हणायचं, मेसोफिल सेल्स. कोणत्या सेल्स असतात? मेसोफिल सेल्स. आणि या मेसोफिल सेल्सच्या मध्ये या ठिकाणी क्लोरोप्लास्ट आहे, यांच्या अलॉंग विथ दुसरा सेल ऑर्गेनिल माहिती आहे आपल्याला, ज्यांना काय म्हणतो आपण? मायटोकॉन्ड्रिया. आणि यांच्यासोबत जो तिसरा सेल ऑर्गेनिल आहे ज्याला काय म्हटलं मी इथं? पर oxysomes. So peroxisomes is the microbodies which is present inside the mesophyll cell along with the chloroplast and mitochondria. Whenever one of the process is completed, that process is known as the photorespiration. वाक्य विसरायचं नाही आहे. मेसोफिल सेल मध्ये या 3 सेल ऑर्गेनिल पासून जी एक प्रोसेस होते त्या प्रोसेसला काय म्हणायचं? फोटोरेस्पायरेशन. So that is known as the peroxisome. Okay, now again the second cell organelle, minute cell organelle, or that is known as the microbodies, which is also known as the glyoxysome. See, this glyoxysome which is present only plant cell and which is responsible for main functions. काय करतं पहा? glyoxysome which is helpful for the present किंवा लोकेशन जर पाहिलं आपण, which is present inside the guard cell. कोणत्या सेल्स मध्ये असतं? गार्ड सेल्स ऑफ स्टोमॅटा. पहा, this glyoxysome which stored the different types of enzymes which is required for the different metabolic activities. आणि ज्यांच्यामध्ये ॲक्च्युअल फॅटी ज्या टिश्यू असतात किंवा फॅट स्टोरेज जो पार्ट्स असतो यांच्यामध्ये हे कोण प्रेझेंट असतात? ग्लायोक्सिझोम. ते एक्झाम्पल लक्षात ठेवा. जर तुम्ही कॅस्टर सीड्स पाहिलं असेल ज्यांच्यामध्ये फॅट्स असतात, त्या फॅट्समध्ये स्टोरेज करणारा पार्ट जो आहे फॅट स्टोरेजिंग, ज्याला काय म्हणतो आम्ही? ग्लायोक्सिझोम. इट ऑल्सो कंटेन्स इनसाइड द गार्ड सेल्स ऑफ. गार्ड सेल्स कुणाचा पार्ट्स आहे? सो गार्ड सेल्स इज द पार्ट्स ऑफ स्टोमॅटा. स्टोमॅटा मध्ये तो प्रेझेंट असतो. पहा, डायग्रॅमॅटिकल जर स्ट्रक्चर पाहिलं आपण, अशा किडनी शेप्ड 2 सेल्स असतात ज्या सराउंडेड मायन्यूट ओपनिंगला करतात आणि या जनरली कुठं प्रेझेंट असतात? अपर अँड लोअर एपिडर्मिस. फॉर एक्झाम्पल इथं ही लोअर एपिडर्मिस आहे. या लोअर एपिडर्मिसमध्ये या ज्या इपिडर्मल सेल्स असतात, ह्या एपिडर्मल सेलचं मॉडिफिकेशन होतं आणि अशा किडनी शेप्ड सेल्स तयार होतात. आणि या 2 सेलला काय म्हणतो आपण? दॅट इज नोन एज द गार्ड सेल्स अँड विच सराउंडेड बाय द एपिडर्मल सेल अँड दॅट स्ट्रक्चर इज नोन एज द. ह्या एंटायर स्ट्रक्चरला काय म्हणतो आम्ही? स्टोमॅटा. So this stomata which having the guard cell, and inside the guard cell the another one microbodies is present. That microbodies is also known as the glyoxysome. Now another one is the speriosomes. So speriosomes is also present inside the plant cell and which perform the different types of metabolic activities due to the presence of enzymes. पहा, लिपिड स्टोरेज आणि लिपिड सिंथेसिससाठी जो मेन सेल ऑर्गेन असतो तो कोणता असतो? स्पेरियोसोम. So all these are the minute microbodies present inside the cytoplasm of plant cell and in some plant and animal cells. So that is called like the microbodies. तर हा पार्ट्स या ठिकाणी आपल्याला लक्षात ठेवायचा आहे. NCERT मध्ये 2 वर्ड दिलेत फक्त. मायक्रोबॉडीज आर द स्मॉल व्हेसिकल्स और विच इज एनक्लोज्ड बाय द डिफरंट टाइप्स ऑफ एन्झाईम, दॅट इज नोन एज द मायक्रोबॉडीज. पण याचा रेफरन्सेस आपल्याला नेक्स्ट टॉपिकमध्ये जिथे जिथे येणार आहे त्यावेळेस आपल्याला तो डिटेल्स पाहायचा आहे. तर त्या ठिकाणी मायक्रोबॉडीज म्हटलं तर स्मॉल व्हेसिक्युलर स्ट्रक्चर सायटोप्लाझममध्ये असतात. तर या ठिकाणी आपला हा एंटायर टॉपिक सेल और युनिट ऑफ लाईफ हा कंप्लीट झालेला आहे. हा टॉपिक पूर्णपणे आपला इथं संपलेला आहे. याच्यानंतरच्या नेक्स्ट व्हिडिओ लेक्चरमध्ये आपण पाहणार आहेत की याच्यावर MCQs कसे विचारले जाणार आहेत. ॲक्च्युअली आता आपण टॉपिक शिकून संपलेला आहे, पण या टॉपिकवर MCQs कसे येतात, नीटला प्रश्न कसे विचारले जातात, तर यांचं डिस्कशन आपण नेक्स्ट व्हिडिओ लेक्चरमध्ये पाहणार आहेत. पण याच्या पूर्वी जो आपण एक व्हिडिओ अटॅच केला होता पहा, ज्याच्यामध्ये सेलची इन्फॉर्मेशन्स होती, यांच्यामध्ये ॲडिशन काही पॉईंट्स करून परत याला आपण एक व्हिडिओ अटॅच करतोय ज्याच्यामध्ये एंटायर सेल सुरुवातीपासून ते शेवटीपर्यंत सर्व कॅरेक्टर त्यामध्ये एनक्लोज्ड केले. तर तो पार्ट्स पाहायचा आहे आणि नेक्स्ट व्हिडिओ लेक्चरमध्ये आपण स्टार्ट करतोय MCQs ऑफ सेल. ओके.
Animal and plant species of varying sizes and shapes inhabit the earth, and they all have one thing in common. They are all made up of cells, the fundamental unit of life in living organisms. Scientists didn't know about the existence of cells until the invention of the microscope. Anton van Leeuwenhoek, an amateur Dutch scientist, is credited as the first person to see and describe living cells. Later, Robert Brown expanded on Leeuwenhoek's studies and pointed to the presence of a cell nucleus. In 1838, German botanist Matthias Schleiden examined numerous plant species and concluded that plants have different types of cells which form plant tissues. A year later, Theodore Schwann, a British zoologist, examined different animal cells and discovered that the cells have a thin outer layer, which we now know as the cell or plasma membrane. He also studied plant cells and concluded that in addition to a cell membrane, plant cells also have a cell wall, a characteristic unique to plants. Schleiden and Schwann together formulated the cell theory, which stated that both plants and animals are made up of cells and their products such as proteins and lipids. However, the cell theory did not explain the formation of new cells, which was clarified by Rudolf Virchow in 1855. He coined the Latin phrase omnis cellula e cellula, which meant that new cells arise from pre-existing cells, and accordingly modified the cell theory, which now states that all living organisms are composed of cells and products of cells, and these cells arise from pre-existing cells. Cell research received a further boost with the invention of the electron microscope, as it enabled scientists to observe the structure of the cell. They observed that the structure of a cell varies in plants and animals. A plant cell, for instance, is surrounded by a distinct cell wall on the outside and a plasma membrane on the inside, whereas an animal cell lacks a cell wall and has a plasma membrane instead that forms the outer cell boundary. Both animal and plant cells have a dense and spherical structure called the nucleus, which contains thread-like structures called chromosomes. Each chromosome is made of deoxyribonucleic acid or DNA, which stores the genetic information of the organism. If the nucleus is membrane-bound, then the cell is called an eukaryotic cell. They are found in members of kingdom Protista, kingdom Fungi, kingdom Plantae, and kingdom Animalia. On the other hand, cells without a membrane-bound nucleus are called prokaryotic cells, and members of kingdom Monera possess such cells. Both eukaryotic and prokaryotic cells are filled with cytoplasm, a semi-fluid matrix present inner to the cell membrane. However, in an eukaryotic cell, the cytoplasm does not include the nucleoplasm, the nucleus matrix filled with the contents of the nucleus. Cytoplasm is a hub of activities and chemical reactions, such as protein synthesis, that help the cell remain alive. Suspended inside the cytoplasm are distinct structures called organelles. In eukaryotic cells, we find membrane-bound organelles such as the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles, as well as non-membrane-bound organelles such as ribosomes. Interestingly, Ribosomes are also found in chloroplasts of plant cells as well as the mitochondria and rough endoplasmic reticulum of both plants and animals. Moreover, animal cells possess another non-membrane bound organelle called the centriole, which aids in cell division. The cytoplasm of prokaryotic cells, on the other hand, lacks membrane-bound organelles but contains ribosomes and fragments of extra-chromosomal DNA called plasmids. Cells usually differ in size and shape. For instance, mycoplasma measure only 0.3 microns in length, whereas human red blood cells are about 7 microns in diameter and nerve cells measure around 4 to 100 microns. Amazingly, the ostrich egg is the largest isolated single cell, and it measures 17 to 19 centimeters in length and 14 to 15 centimeters in width. Moreover, the shapes of cells range from disc-like to thread-like and from polygonal to columnar and cuboid. Today, though we have gained a lot of information about cells, scientists are still engaged in unlocking the secrets of the cell and its organelles. Millions of microorganisms are present in every nook and corner of our surroundings. Of these, certain microorganisms, such as bacteria, have a nucleoid but no membrane-bound organelles in their cytoplasm. Such organisms are called prokaryotes. Prokaryotes are smaller and multiply more rapidly than eukaryotes. All members of kingdom Monera, including bacteria, blue-green algae, and pleuropneumonia-like organisms or mycoplasma, are prokaryotes. And they can be rod-shaped, spherical, spiral-shaped, or comma-shaped. Microscopic studies reveal that most prokaryotes, except for a few such as mycoplasma and thermoplasma, possess a cell envelope, a complex, tightly bound, three-layered chemical structure consisting of the outermost glycocalyx, followed by the cell wall and then the plasma membrane. The layers together act as a single protective unit, though each has its distinct function. The glycocalyx, for instance, helps prokaryotes such as bacteria to resist antibiotics when they invade host cells. Did you know that the glycocalyx differs in thickness and composition among different prokaryotes? In some, it occurs as a thick and tough sheath called a capsule, while in others it may be a loose sheath called a slime layer. The glycocalyx is followed by a cell wall, which determines the shape of the cell and provides structural support, thereby preventing the cell from collapsing or bursting. Interestingly, the cell wall structure differs in different prokaryotes. In certain prokaryotes, such as Pseudomonas, the cell wall is composed of a thin inner layer of peptidoglycan and an outer membrane of lipopolysaccharide and lipoprotein. When such prokaryotes are subjected to the Gram staining procedure, they do not take up the Gram stain, but appear red or pink when counterstains such as safranine are added. Hence, they are called gram-negative bacteria. On the other hand, the cell wall of prokaryotes such as Bacillus anthracis, composed of a dense layer of peptidoglycan and teichoic acids, turns violet when subjected to the gram staining procedure. Hence, such prokaryotes are called gram-positive. The cell wall is followed by the semi-permeable plasma membrane, which controls the entry and exit of small molecules like food particles and ions like chloride, sodium, and bicarbonates. Often, the plasma membrane extends into the cell, forming the mesosome, a membranous structure seen in the form of vesicles, tubules, and lamellae. Incidentally, the presence of the mesosome is a characteristic feature of prokaryotes. Mesosomes help in cell wall formation, respiration, secretion processes, expansion of the surface area of the plasma membrane and enzymatic content, DNA replication, and distribution to daughter cells. Some prokaryotes, such as blue-green algae, also have membranous extensions called photosynthetic lamellae that contain photosynthetic pigments such as chlorophyll. Prokaryotes also have extensions from their cell walls. For instance, motile bacterial cells have a thin filamentous extension from their cell wall called a flagellum, which consists of a filament, hook, and a basal body, with the filament being the longest portion. Motile as well as non-motile prokaryotes also possess pili and fimbria, surface structures that attach the prokaryotes to rocks in streams and also to host tissues. While pili are elongated tubular structures made up of a special protein called pilin, fimbria are tiny bristle-like fibers sprouting out of the cell. However, these structures don't play any role in motility. While pili and fimbria are present outside the cell envelope, the inside is filled with cytoplasm that hosts the genomic DNA, a single chromosome made up of circular DNA. Many prokaryotes also have small circular DNA called plasmids outside their genomic DNA, which confer unique enotypic characteristics such as resistance to antibiotics. The cytoplasm also contains ribosomes measuring about 15 microns by 20 microns in size, and consisting of two subunits, 50S and 30S, which together form the 70S ribosome. Here, S, or Svedberg's unit, stands for the sedimentation coefficient. It indirectly is a measure of the density and size of ribosomes. These ribosomes attach to a single messenger RNA, a type of RNA that aids in protein synthesis, forming a chain called a polyribosome or polysome, and ultimately translate messenger RNA into proteins. Apart from the ribosomes, Cytoplasm also contains inclusion bodies such as phosphate granules, synophycean granules, and glycogen granules, which are used for storing reserve materials. These bodies are not bounded by any membrane system and lie free in the cytoplasm. Certain prokaryotes such as blue-green, purple, and green photosynthetic bacteria also have other inclusion bodies such as gas vacuoles, which help them to float in water. Prokaryotes, thus, are primitive organisms that possess a simple cell structure. Yet, they are the only organisms to thrive in almost all habitats, including underground caves and deep oceans. Our Earth is home to diverse species of plants and animals. Yet, they all have a common structural unit: the eukaryotic cell, or a cell which possesses an organized nucleus surrounded by a nuclear envelope. This nucleus, containing chromosomes that carry the genetic code of the organism, is surrounded by cytoplasm, a semi-fluid matrix which makes up the volume of the cell. Present in the cytoplasm is the cytoskeleton, an elaborate network of filamentous, proteinaceous structures that gives the cell its characteristic shape, aids in intracellular transport, gives mechanical strength to the cell, and also helps in motility in case of motile cells. The cytoplasm also contains numerous single membrane-bound minute vesicles called microbodies, which are filled with enzymes that aid in the synthesis of amino acids. Also embedded in the cytoplasm are several organelles or membrane-bound structures which lead to its extensive compartmentalization. However, the cytoplasm of all eukaryotic cells does not contain the same set of organelles. Plant cells, for instance, have a large central vacuole and plastids, whereas centrosomes are only found in animal cells. The centrosome, surrounded by amorphous pericentriolar material, is made up of two cylindrical structures called centrioles that lie perpendicular to each other. Each centriole is made up of 9 evenly spaced peripheral fibrils of tubulin protein, with each peripheral fibril being a triplet. All adjacent triplets are linked to each other, forming a cartwheel-like structure. The central part of the centriole, called the hub, is proteinaceous and is linked to the tubules of the peripheral triplets by radial spokes made of protein. Centrioles form the basal body of cilia or flagella and spindle fibers that give rise to a spindle-like apparatus during cell division. Eukaryotic cells differ not only in their cytoplasmic organelles, but also in their structure. Take, for instance, animal cells in which the plasma membrane forms the outer boundary of the cell. But in fungi and plant cells, the plasma membrane is covered by a cell wall, which forms the outer boundary. Interestingly, the composition of the cell wall is different in different eukaryotic cells. In algae, the cell wall is made up of cellulose, mannans, galactans, and calcium carbonate, whereas in fungi, it is made up of chitin and glucans. On the other hand, a plant cell wall is made up of cellulose, hemicellulose, pectins and proteins, consists of 3 layers: the middle lamella, primary wall and secondary wall. The middle lamella, mainly made of calcium pectate, lies between neighboring cells and glues the cells together. The primary wall is the first formed wall that develops when the cell is growing. It is thin, elastic and extensible. However, its growth capability diminishes as the cell matures. The secondary cell wall develops after the cell has matured. It is formed on the inner side of the cell and faces the plasma membrane. Interestingly, the secondary cell wall and lamellae do not occur as continuous layers, as they are interrupted by plasmodesmata, fine strands of cytoplasm that facilitate the transportation of materials between different cells in a plant. The cell wall is an important part of the cell as it lends shape to the cell, protects it from mechanical damage and infection, prevents the entry of undesirable macromolecules such as nucleic acids, and also aids in cell-to-cell interaction. Though a cell wall may be present or absent in different types of eukaryotic cells, these cells are the basic unit of life in many organisms, including plants and animals. The cell, the basic unit of life in all organisms, has several organelles embedded in its cytoplasm. These organelles are surrounded by a thin membrane called the plasma membrane, which not only separates the extracellular environment from the cytoplasm, but also gives shape to the cell. Before the advent of the electron microscope, scientists conducted chemical studies on the cell membrane and deduced that it is composed of lipids that mainly consist of phosphoglycerides. These lipids are arranged in a bilayer with the polar hydrophilic head of both layers pointing outward towards the aqueous environment, and the nonpolar hydrophobic tail made of saturated hydrocarbons pointing inward. Such an arrangement not only protects the tail from the aqueous environment, it also prevents polar molecules such as nucleic acids, proteins and ions from diffusing through it. Later, biochemical investigations revealed that apart from lipids, proteins and carbohydrates too are a part of the cell membrane. Moreover, the proteins are arranged in 2 forms, peripheral and intrinsic. Peripheral proteins lie on the surface of the membrane and can be easily removed by physical methods. On the other hand, intrinsic proteins are totally or partially embedded in the bilayer and can be removed only after disrupting the entire membrane. Scientists also deduced that the ratio of proteins to lipids varies greatly between different types of cells. For instance, the cell membrane of red blood cells is 52% proteins and 40% lipids. The advent of the electron microscope helped scientists better understand the structure of the cell membrane, and finally in 1972, Singer and Nicholson put forth the fluid mosaic model, which was widely accepted by the scientific community. This model stated that lipids are found in the form of a fluid bilayer. This quasi-fluid of almost fluid-like nature allows proteins to move laterally within the bilayer, and this ability of the membrane to allow the movement of molecules within it is referred to as membrane fluidity. The fluid nature of the membrane also allows the cell to perform important functions such as cell growth, the formation of intercellular junctions, secretion, and cell division. All these functions require flexibility in the membrane. For instance, during cell growth and cell division, the membrane has to extend. The cell membrane also guards the entry and exit of different molecules and ions in and out of the cell. However, the cell membrane is selectively permeable, which means the lipid bilayer allows only certain small non-polar substances such as oxygen and carbon dioxide molecules to pass freely through it, while selectively permitting ions and other polar molecules, including water, to pass through it with the aid of membrane proteins. The transport of these ions and molecules takes place either through passive transport or through active transport. In passive transport, substances require no metabolic energy to move across the membrane. Passive transport occurs either through diffusion or osmosis. In a cell membrane, we observe simple diffusion and facilitated diffusion. In simple diffusion, oxygen and carbon dioxide molecules freely move across the membrane from higher to lower concentration, whereas larger molecules, such as glucose, are transported by facilitated diffusion, in which carrier proteins in the membrane change their shape and accelerate the diffusion process. On the other hand, osmosis refers to the movement of water molecules across the cell membrane through diffusion. In active transport, transmembrane proteins act as pumps and utilize the energy from ATP to move substances against their concentration gradient, that is, from lower concentration to higher concentration. An example for this is the sodium-potassium pump. Sometimes, glucose is also transported against the concentration gradient via active transport. The cell membrane, which helps in the transport of substances, is also a part of the cilia and flagella, hair-like structures that project from the cell's surface. These structures emerge from centriole-like structures called basal bodies and share a similar structure, though cilia are smaller than flagella. Their core, called the axoneme, is covered by the cell membrane and has numerous microtubules running parallel to its axis. Two microtubules join to form a doublet. If we observe a cross-section of the axoneme, we will see 9 pairs of radially arranged peripheral doublets and a pair of centrally located microtubules which are interconnected by a bridge. Such an arrangement of axonemal microtubules is known as the 9+2 array. The central microtubules are enclosed by a central sheath, which is connected to one of the microtubules of each peripheral doublet by a radial spoke. The peripheral doublets too are interconnected by linkers called an interdoublet bridge. The cilia and flagella help the cell propel or move the fluid surrounding the cell. Thus, it helps in motility. Thus, cilia and flagella help cells in motility, while the cell membrane gives shape to the cell, separates the intracellular components from the extracellular matrix, and also plays an important role in the transport of substances across the cell. The cytoplasm of a eukaryotic cell is embedded with several membranous organelles, of which the endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles are referred to as an endomembrane system because their functions are coordinated. The endoplasmic reticulum, or ER, for instance, synthesizes, processes, and transports proteins and lipids to the other organelles, as well as metabolizes carbohydrates in liver cells and detoxifies drugs present in the body. Microscopic studies reveal that the endoplasmic reticulum occurs as a network or reticulum of tiny tubular structures dividing the intracellular space into two distinct compartments. The luminal compartment, which consists of the space within the folds of the endoplasmic reticulum, and the extraluminal compartment, which consists of the cytoplasm that lies outside the folds. There are two types of endoplasmic reticulum: rough endoplasmic reticulum, or RER, and smooth endoplasmic reticulum, or SER. The rough endoplasmic reticulum has ribosomes attached to its surface and is mainly found in cells actively involved in protein synthesis and secretion. They are continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum, on the other hand, is devoid of ribosomes and hence appears smooth. It synthesizes lipids and lipid-like steroidal hormones such as estrogens in the case of animal cells. Did you know that the smooth endoplasmic reticulum present in muscle cells is called the sarcoplasmic reticulum, and that it stores and pumps calcium ions? Apart from the endoplasmic reticulum, the endomembrane system also consists of the Golgi apparatus. Named after Camillo Golgi, it consists of a series of flat, disc-shaped sacs or cisternae stacked one above the other. Each cisterna measures 0.5 to 1 micron in diameter, and their number may vary in each Golgi apparatus. The cisternae, concentrically arranged near the nucleus, have two distinct faces: the convex-shaped cis, or the forming face that faces the nuclear envelope, and the concave-shaped trans, or the maturing face that lies opposite the nuclear envelope. Though both the cis face and the trans face are interconnected, they are different. For instance, the cis face has a thin membrane, whereas the membrane of the trans face is thick. In a cell, the Golgi apparatus acts as a packaging and forwarding center. For instance, vesicles containing proteins synthesized by the ribosomes of the rough endoplasmic reticulum get pinched off the surface of the rough endoplasmic reticulum and fuse with the cis face of the Golgi apparatus. Here they are modified in the cisternae before they are released from the trans face. This explains why the Golgi apparatus remains closely associated with the endoplasmic reticulum. In addition to being a packaging and forwarding center, the Golgi apparatus is an important site for the formation of glycoproteins and glycolipids that form a part of the plasma membrane. Lysosomes, another member of the endomembrane system, are single-membrane vesicular structures formed during the packaging process in the Golgi apparatus. They contain hydrolytic enzymes such as lipases, proteases, and carbohydrases that digest lipids, proteins, and carbohydrates, and also nucleic acids. Apart from lysosomes, the endomembrane system also comprises vacuoles. Enveloped in a membrane called tonoplast, a vacuole contains water, sap, excretory products, and other materials not useful for the cell. The tonoplast of vacuoles in plant cells aids in the transportation of a number of ions and other materials against their concentration gradients into the vacuole, which is one of the reasons for the higher concentration of ions in the vacuole. In plant cells, vacuoles can make up 90% of the volume of the cell. These vacuoles also perform different functions. For instance, in amoeba, the contractile vacuole helps the organism excrete waste products and also in osmoregulation. On the other hand, in protists, food vacuoles formed by engulfing food particles provide nutrition to the organism. All the organelles of the endomembrane system work in a coordinated manner and play a vital role in biosynthesis, processing, packaging, transport, and secretion of proteins and lipids, as well as in storage and digestion of wastes. In 1953, George Pallard was observing a cell under an electron microscope when he noticed a few dense particles in the cytoplasm of the cell. Today, we know these particles as ribosomes. These granular and non-membrane bound structures, composed of ribosomal RNA and proteins, are found in both prokaryotic and eukaryotic cells. They synthesize proteins by attaching to a single messenger RNA to form a chain called a polyribosome or polysome, and ultimately translate mRNA into proteins in both eukaryotes and prokaryotes. While eukaryotic ribosomes are 80S and composed of 40S and 60S subunits, prokaryotic ribosomes are 70S and composed of two subunits, 30S and 50S. Apart from the cytoplasm, ribosomes also occur on the rough endoplasmic reticulum, in chloroplasts in plants, as well as within the mitochondria. Called the powerhouse of the cell, mitochondria are sites of aerobic respiration and produce cellular energy in the form of ATP. The number of mitochondria in a cell varies depending on the cell's metabolism. Muscle cells, for instance, need a continuous supply of energy, and hence they contain a large number of mitochondria. Microscopic studies have revealed that a typical mitochondrion, which divides by fission, is sausage-shaped or cylindrical, with a diameter of 0.2 to 1 microns and a length of 1 to 4.1 microns. It is a double membrane bound structure consisting of an outer membrane and an inner membrane, and each membrane has its own specific enzymes associated with the mitochondrial function. The membranes divide the mitochondrial lumen into two aqueous compartments, the outer compartment and the inner compartment called the matrix, that contains a single circular DNA molecule, a few RNA molecules, 70S ribosomes, and the components required for the synthesis of proteins. While the outer membrane forms a continuous limiting boundary, the inner membrane folds towards the matrix to form infoldings called cristae. These cristae increase the surface area of the inner membrane of the mitochondria. Apart from ribosomes and mitochondria, plastids are also one of the most important cytoplasmic organelles in plants. They are bound by a double membrane and contain pigments that impart specific colors to plants. Based on the pigments they possess, plastids can be classified as chloroplasts, chromoplasts, and leucoplasts. Chloroplasts contain chlorophyll and carotenoid pigments that capture light energy needed for photosynthesis. Moreover, chlorophyll also imparts a green color to the leaves. Found mostly in the mesophyll cells of the leaves, chloroplasts have a length of 5 to 10 millimeters and a width of 2 to 4 millimeters. A cell can contain a single chloroplast, as seen in Chlamydomonas, or multiple chloroplasts, as seen in a green alga cell. Morphological studies about chloroplast reveal that they possess an outer membrane and an inner membrane, with the inner membrane being less permeable than the outer membrane. Stroma, the space enclosed by the inner membrane, contains double-stranded circular DNA molecules, 70S ribosomes, and enzymes required for the synthesis of carbohydrates and proteins. Stroma also contains thylakoids, flattened membranous sacs containing chlorophyll pigment in their lumen, the space enclosed by the thylakoid membranes. Around 20 to 50 thylakoids are stacked like piles of coins, forming grana or intergranal thylakoids. The thylakoids of different grana are connected to each other by flat membranous tubules called stroma lamellae. Did you know that chloroplasts as well as mitochondria are known as semi-autonomous organelles as they possess their own DNA? Chromoplasts, another type of plastids found in plant cells, contain fat-soluble carotenoid pigments such as carotene and xanthophylls that give red, yellow, and orange color to plant parts. The third type of plastids, leucoplasts, are colorless plastids that store nutrients such as carbohydrates and fats. Based on the type of nutrient stored, leucoplasts can be classified as amyloplasts, eleoplasts, and aleuroplasts. While amyloplasts store carbohydrates such as starch in potato, eleoplasts store oils and fats. On the other hand, aleuroplasts, which are usually found in food grains, store proteins. The various plastids found in plants not only impart color to the plants, but also store nutrients and play a role in photosynthesis. Thus we find that ribosomes, mitochondria, and plastids are important organelles taking part in vital functions of the cell, such as protein synthesis, respiration, and photosynthesis. Of the various organelles found in a cell, the nucleus occupies a place of prime importance as it is the control center that regulates metabolic activities which keep a cell alive. Most eukaryotic cells possess a single nucleus. However, we do find cells with multiple nuclei as well as cells without a nucleus. Described by Robert Brown in 1831, the nucleus was one of the earliest organelles that scientists became aware of. The advent of the electron microscope enabled scientists to become aware of the various components of a nucleus. For instance, scientists observed that the nucleus is surrounded by a nuclear envelope or nuclear membrane that acts as a barrier between the contents of the nucleus and the other organelles present in the cytoplasm. The nuclear envelope consists of the outer membrane and inner membrane, which run parallel to each other. and are separated by the perinuclear space. The outer membrane of the nuclear envelope is connected with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the two membranes fuse and give rise to minute pores called nuclear pores. These pores allow two-way movement of RNA and protein molecules between the nucleus and the cytoplasm. Present inside the nuclear envelope is the nucleoplasm or the nuclear matrix that contains a spherical structure called the nucleolus. The nucleolus lacks a membrane which makes its content continuous with the nucleoplasm. The nucleolus is a major site for active ribosomal RNA synthesis, and hence cells that are active in protein synthesis have larger and numerous nucleoli. Apart from the nucleolus, the nucleoplasm also contains chromatin, a network of highly extended and elaborate nucleoprotein fibers that mainly consists of DNA and histone proteins, though non-histone proteins are also associated with it. When a cell begins to divide, the chromatin condenses and coils to form rod-like structures called chromosomes. The DNA threads remain distributed in these chromosomes. Morphological studies of a chromosome have revealed that it consists of a primary constriction or centromere that has disc-shaped structures called kinetochores attached to its side. The parts present on either side of the centromere are called the arms of the chromosomes. Based on the position of the centromere, chromosomes can be classified as metacentric, submetacentric, acrocentric, and telocentric. In metacentric chromosomes, the centromere is exactly in the middle, forming two equal arms. In submetacentric chromosome, the centromere appears closer to one arm of the chromosome, creating two arms of unequal length. An acrocentric chromosome has its centromere located very close to one of its ends, resulting in one extremely short and one very long arm. In telocentric chromosomes, the centromere is located at one end of the single arm. At times, chromosomes have secondary constrictions at a fixed location, lending the appearance of a small fragment called a satellite. These areas of secondary constriction help in the formation of a nucleolus after cell division. Chromosomes are an important part of the nucleus as they contain the genetic code of an organism. Thus, we have seen that the nucleus, along with its components, is an important organelle as it not only coordinates the functioning of the cell but also plays a vital role in cell division.